there is a moment that every soldier knows. It comes somewhere between the second and third hour of a forced march in extreme heat, when the body begins to betray its owner. The legs grow heavy. The eyes lose their sharpness. The mouth turns to sand. Decisions that should take seconds begin to take minutes. And the water in the canteen, despite having consumed it all, offered far less protection than the soldier was promised. This is not a story of weakness. This is a story of science that was hidden in plain sight for decades, quietly deployed on battlefields and in military training facilities around the world, while the rest of humanity kept drinking plain water and wondering why it was never quite enough. What follows is the true account of how a handful of army researchers, wartime physicians, and biochemists cracked one of the most fundamental problems in human physiology: how to make the body absorb water up to 3 times more efficiently, and how that discovery, worth billions of dollars to the sports drink industry, can be replicated by any person on the planet for approximately $16. The human body is composed of between 50 and 70% water. That figure is not metaphorical. It is structural. Water is the medium through which every chemical reaction in the body occurs. It regulates body temperature, carries oxygen through the bloodstream, lubricates joints, protects organs, and maintains the electrochemical balance that allows neurons to fire. Without water, the machinery of human life simply cannot run. And yet, for most of recorded history, the conventional wisdom about hydration was profoundly incomplete. Armies marched on it. Athletes trained on it. Entire medical systems were built on it. The assumption was simple: when you are thirsty, drink water. When you sweat, replace what you lost with more water. The problem is that this assumption is wrong, or rather, it is dangerously incomplete. During World War II, the United States military began to confront a problem that was killing soldiers not from bullets, but from the sun. In the deserts of North Africa and the Pacific Islands, troops were falling victim to heat exhaustion and heat stroke in numbers that alarmed military commanders. The standard response was to order soldiers to drink more water. It helped, but not nearly enough. Soldiers who drank enormous quantities of water were still collapsing. Something was missing from the equation, and nobody at the time fully understood what it was. The investigation that followed would take nearly 2 decades and would ultimately upend everything the medical community believed about how the human body absorbs fluids. The first breakthrough came not from a general or a military strategist, but from a physiologist studying the intestinal lining of laboratory rats. In 1960, a biochemist named Robert Crane published findings that would eventually be recognized as one of the most important discoveries in the history of medicine. Working at the cellular level, Crane identified a transport mechanism in the small intestine that had never been properly described before. He found that sodium, the primary salt in the human body, could not efficiently cross the intestinal wall into the bloodstream on its own. It needed a partner. And that partner was glucose, a simple sugar. The mechanism Crane discovered is now known as the sodium-glucose cotransport system, carried out by a protein called SGLT1. Here is how it works. When sodium and glucose are present in the small intestine at the correct ratio, a molecular pump embedded in the intestinal wall activates. It grabs 2 sodium ions and 1 glucose molecule simultaneously and carries them together across the cell membrane and into the bloodstream. And critically, water follows. For every cycle of this transport mechanism, hundreds of water molecules are pulled across the intestinal wall by osmotic pressure, flooding into the bloodstream with a speed and efficiency that plain water cannot match. The implications were staggering. It meant that the body was not simply a vessel that absorbed water passively. It meant that hydration was an active, chemistry-dependent process, and that without the correct chemical key, the door to true hydration remained largely locked. This discovery would not reach military or public health applications immediately. Science moves slowly and paradigms resist change. But the research trail had been opened, and it was about to lead somewhere extraordinary. The first clinical application came in 1964, when United States Army Captain Robert Phillips, working in the Philippines, documented the first successful use of a glucose saline solution on cholera patients. Cholera kills through dehydration with terrifying speed. The infected body loses fluid so rapidly that death can occur within hours. Physicians had long struggled to keep patients alive long enough for the infection to pass, relying on intravenous fluid therapy administered through needles in hospital settings. It was expensive, logistically complex, and entirely unavailable in field conditions or developing world clinics. Phillips' glucose saline solution changed that. By combining the correct amounts of sodium and glucose in water, he created a drink that activated the SGLT1 cotransport system and allowed the body to absorb fluids at a rate that came close to, and in some contexts matched, the effectiveness of N4 drip. A patient could drink this solution and rehydrate themselves without a needle, without a hospital, and without a doctor in the room. Shortly after, researchers at the Infectious Disease Hospital in Calcutta and the Cholera Research Laboratory in Dhaka refined the formula further, producing what would become the first modern oral rehydration solution. When tested on cholera patients, the results were so dramatic that the medical community initially struggled to believe them. The real proof came on a battlefield, not the one the researchers had anticipated. In 1971, during the Bangladesh Liberation War, a cholera epidemic swept through refugee camps holding millions of displaced civilians. Medical resources were nearly non-existent. Fluid therapy was impossible at the scale required. People were dying in the thousands. A small team of physicians, led by researcher Dilip Mahalanabis, made a decision that was considered radical at the time. They mass-produced the oral rehydration solution, gave it to camp workers with minimal training, and instructed families to administer it to sick relatives themselves. The formula was elegant in its simplicity, the correct ratio of sodium, glucose, potassium, and clean water. The mortality rate dropped from 30% to less than 4%. That number, 30% to less than 4%, represents one of the most dramatic reductions in mortality ever recorded in a medical intervention. The Lancet, one of the world's leading medical journals, later described oral rehydration therapy as potentially the most important medical advance of the 20th century. The World Health Organization formalized the formula in the 1970s and distributed it globally. By 2003, WHO updated the specifications to reduce the osmolarity of the solution, making absorption even more efficient. Studies published in JAMA and Cochrane databases confirmed that the reduced osmolarity formula decreased the duration of dehydration illness by measurable percentages and reduced the need for IV therapy by approximately 30% compared to earlier versions. The science was not classified. But somehow, it never fully escaped the walls of academic medicine and military nutrition research. Meanwhile, the United States military was learning a parallel set of lessons about hydration in non-disease contexts. Specifically about what happens to a soldier's body and mind when they lose water under the stress of combat training. The findings were alarming. Research commissioned by the U.S. Army Research Institute of Environmental Medicine demonstrated that cognitive performance, the ability to think, decide, aim, and react, begins to degrade when a soldier loses as little as 2% of their body mass to dehydration. That is not a large number. For a soldier weighing 80 kilograms, 2% represents 1.6 liters of fluid, an amount that can be lost within the first hour of moderate exertion in the heat. At that threshold, attention falters. Reaction time slows. Short-term memory begins to collapse. A study published in the British Journal of Nutrition confirmed that dehydration of just 2% impairs performance in tasks requiring attention, psychomotor coordination, and immediate memory recall. A separate study examined 31 elite U.S. Army officers during a 53-hour simulated combat exercise conducted in heat. The results were sobering. The soldiers slept an average of 3 hours over the entire exercise. They lost an average of 3 liters of water through sweat alone. The degradation in cognitive function, measured through computerized testing, was described by researchers as substantial. Decision-making, reaction time, and working memory all declined measurably. Military pilots were found to be particularly vulnerable. Research published in Military Medicine demonstrated that dehydrated pilots performed significantly worse on spatial cognition and flight performance tests compared to hydrated pilots. The implications for flight safety were, in the words of the researchers, serious. The Defense Health Agency reported that in 2020 alone, more than 2,000 troops were afflicted with heat exhaustion. 20% of heat-related military illnesses occur outside the traditional summer heat season, meaning soldiers were falling in the cold, in the fall, in the spring, because the relationship between hydration and environmental conditions is more complex than temperature alone. The military understood something that the general public did not. Dehydration is not primarily a thirst problem. It is a performance problem. It is a cognition problem. It is, under extreme conditions, a survival problem. And plain water, consumed alone, is not the answer. Here is the biology that changes everything, explained simply. When a person drinks plain water, it enters the stomach and passes into the small intestine. From there, it must cross the intestinal wall and enter the bloodstream. This process relies in part on passive osmosis, a slow, imprecise mechanism that becomes even less efficient when the body is already depleted of electrolytes from sweat. When a person sweats heavily, they do not just lose water. They lose sodium, potassium, magnesium, chloride, and zinc, the electrolytes that maintain the electrical charge of cells, regulate blood pressure, enable muscle contractions, and govern virtually every function of the nervous system. Drinking plain water after significant sweat loss does not restore this balance. In fact, flooding a sodium-depleted system with sodium-free water can dilute the electrolyte concentration in the blood even further, a condition that has its own clinical name, hyponatremia, and which in severe cases can cause seizures, cerebral edema, and death. The sodium-glucose cotransport system solves this problem at the molecular level. By consuming a solution that contains sodium, glucose, and other electrolytes at the precise ratio that activates SGLT1, the body can absorb fluids and replenish electrolytes 2 to 3 times more efficiently than with plain water. The military's nutrition researchers confirmed this. The right ratio of sodium and glucose increases water absorption dramatically into the bloodstream via the cotransport mechanism. Blood volume is restored. Muscle function is preserved. Brain cells, which begin to shrink when dehydrated, return to normal. The heart, which works harder when blood volume drops, can ease its labor. This is not a supplement. This is not a performance-enhancing drug. This is the exploitation of a biological mechanism that evolution built into every human intestine. The only question is whether a person chooses to activate it. The sports drink industry understood this science decades ago. Gatorade, born in 1965 from research at the University of Florida, was built on a crude approximation of the cotransport principle. Its formula combined water, sodium, and carbohydrates specifically to leverage the sodium-glucose mechanism. The product made the Gatorade Corporation billions of dollars. It also introduced a problem. Commercial sports drinks optimize for taste and marketability, not for the precise electrolyte ratio that maximizes absorption. Most sports drinks contain too much sugar and too little sodium to optimally activate the SGLT1 cotransport system. The Merck Manual, the standard medical reference used by physicians worldwide, explicitly states that sports drinks, sodas, and fruit juices do not meet the criteria for effective rehydration and should not be used as substitutes. The solution recommended by the World Health Organization and validated by decades of clinical research in Bangladesh, the Philippines, Egypt, and refugee camps across the developing world is far simpler, far cheaper, and far more effective. An oral rehydration solution, a true one built to WHO specifications, can be assembled from ingredients available at any pharmacy or grocery store. The formula requires sodium chloride, commonly sold as table salt, potassium chloride, available in low-sodium salt substitutes, sodium citrate or baking soda, and glucose in the form of table sugar or dextrose. Mixed with 1 liter of clean water in the correct proportions, the total cost of materials is approximately $16 for a supply that lasts for weeks or months. The Army's own nutrition guidance confirms the principle in less technical language. Drinking small amounts frequently is more effective than large amounts occasionally. Urine should be pale yellow. Clear urine signals that electrolytes are being diluted and flushed, not retained. Caffeinated beverages and alcohol actively pull water from the body. Sodium, paired with a modest amount of glucose and consumed before, during, and after exertion, is the non-negotiable foundation of real hydration. The story of oral rehydration is, in a meaningful sense, a story about the gap between what science knows and what the public hears. The research that led to the SGLT1 discovery, the Bangladesh field trials, the WHO formula, and the military hydration protocols has never been secret. It sits in peer-reviewed journals, military medical manuals, and public health databases, accessible to anyone with an internet connection. The Lancet called it the century's most important medical advance. Global health organizations have credited it with preventing more than 1 million deaths per year from diarrheal illness in developing countries. And yet, in wealthy countries, people continue to reach for expensive sports drinks engineered for taste over function, or for plain water that activates none of the body's most efficient absorption mechanisms, or for caffeinated beverages that actively accelerate the dehydration they are meant to prevent. The soldier carrying 70 pounds of gear through a desert at noon is not superhuman. The endurance athlete who finishes a race in conditions that would hospitalize an untrained civilian is not simply more disciplined. They have been given, or have sought out, a piece of practical physiology that most people never encounter. The science is not complicated. The solution costs less than a cup of coffee. The mechanism is built into every human body by default, waiting to be activated by the correct ratio of sodium and glucose. A precise pinch of salt, a small amount of sugar, a liter of clean water, and the knowledge of what happens when they meet at the wall of the small intestine. That is the entirety of the secret. Not classified. Not proprietary. Not expensive. Just science, finally escaping the walls where it was kept.